Right. Well, yes. So we're interested in trying to analyze multi-environment trial data, and there's this procedure that's been developed over time that has some very practical benefits to it. It's called a StageWise or a two-stage analysis. It's not an idea I came up with. That's just a convenient name I borrowed for the software. You may notice, like I'm fond of taking existing names and then just appropriating them for the software packages I write, like R Appl, right? I didn't make up that name. That was the name of the method, but it helps to promote your brand. So anyways, that's what I'm doing here. There's two-stage analysis has different variations that you'll see. Really the one that's kind of vetted by the biometrics community that I would encourage you all to use is the version that you see described here. In stage one, you're going to analyze each environment separately. And you're going to compute an estimate of the genotype in that environment, and we're going to treat it as a fixed effect. So that's what the acronym BLUE is referring to. It's the best estimate of that genotype in that environment. And that becomes the response variable for your stage two, which is now the analysis over multiple environments. And now you bring into the equation the marker data to compute a blob over all those environments. And you can predict properties of individuals. Those are like breeding values, or you can predict properties of markers like additive marker effects. And this formalism has the benefit that allows you to easily adopt statistical models that can account for the nuances of every environment. So you have different experimental designs, or maybe you have spatial variation. It's very difficult to do all that in an optimal way if you're trying to analyze all the environments together in one shot, which is what a single stage analysis refers to. But in this process, when you compute these blues in each environment, that boils everything down to a single number for that genotype, and you lose the information about what the uncertainty or the standard error of that estimate is. And really, for best practice, you should try and propagate that information into the stage two. That's considered best practice, and make people are aware of that. But you can also not only account for the standard errors, you can also account for covariances between the blues for the different genotypes. And for example, in a model that has spatial variation, that induces covariances, and especially when you start talking about multiple traits, you need to account for covariances. So this a two-stage method that accounts for not just the standard errors, but also the covariances between the blues has been called fully efficient by the biometrics community. And this idea has been developed very nicely by the lab of Hans-Peter Pifo over a long time. So that was the motivation for me to develop a package that would make this fully efficient two-stage method more widely available. And the paper is finally accepted for publication in TAG, and you can see the preprint on bioRxiv in the meantime. And as the title suggests, I've got some other goodies baked into this paper besides the two-stage part of it. It's also got the first time complete model of directional dominance in polyploids. I'm going to go through that a little bit here with you today. So there's the link to the software. The flow diagram gives you an idea of how the software works. Starting at the left-hand side, in stage one, you're analyzing each environment to get those blues and their errors. Then if you have marker data, that gets read in with this read geno function, and together they come together in the stage two function, which is the REML calculation of the variance components. You can summarize those results with the summary command, and then transfer those estimates of the variance components to the BLUP part of the analysis. Not everyone realizes that when we talk about BLUP, technically BLUP actually assumes that the variances are known. Usually we talk about BLUPs, we're really doing two things at one time. We're first estimating the variances, and then we're computing these best predictors conditional on the variances that we've estimated from the same data set. Technically, that's called an empirical BLUP, but that's not a distinction that I'm going to emphasize. I'm just going to be talking about BLUPs. So we have the BLUP in two parts. There's this BLUP prep command, which does the computational heavy lifting. And that allows us to rapidly compute different kinds of blubs, like I was alluding to earlier, whether you're talking about a blub of an individual or a blub for marker. Once you have done the expensive linear algebra, you can generate all those different variations quickly, and that's why I designed the packages. So there's 3 vignettes on the GitHub site, and I went through this a little bit a year ago, so I'm going to, again, doing kind of an abbreviated version and highlighting what's new. But the first vignette talks about a single trait analysis with a homogeneous G by E, or the technical term would be compound symmetry. It assumes that there's a single correlation between all environments given by the variance components. Then we can go to a more complex G by E model, or what I'll say heterogeneous, where layered on top of this homogeneous G by E is another G by E, another kind of G by E for a different factor. And the most common application would be locations in a plant breeding context, but there's no, from a mathematical point of view, it could be management or other types of things that you might be interested in. But I'm going to talk about it like it's location. And it allows for, the software then allows for different potential correlations between different locations, and uses this, what's called the second order factor analytic model to try and do that estimation. And then the third vignette goes back to the more simple G by E model because it introduces the complexity on the trait side as opposed to on the location side, and that's this multiple trait model. So the main updates since last year, there have been some slight changes in the syntax. So if you watch the video from last year and you start to use it, you'll see there are some nuances, but it's all documented in the vignette. And I think I had good reasons for making the changes that I did compared to a year ago. You can now combine marker and pedigree information into this H matrix. And we heard a bit about that from Tim, I think, yesterday in the strawberry example. So that's a feature that I've gotten multiple requests for, so that's why it's there. And then it has this directional dominance model, which I'll talk about. It also does a GWAS. That wasn't really the main goal, but as I became aware of, there's been a new method for GWAS in the last 5-6 years. People have shown that you can actually compute by blub the same kind of statistic that you compute normally as a fixed effect in the traditional mixed model approach for GWAS. And that allows us to actually do a fully efficient 2-stage GWAS, which I don't think any other software can let you do that. So GWAS Poly, for example, in GWAS Poly, if you do a 2-stage analysis, you have to do your stage 1 with some other software, and then you can give your estimates to GWAS Poly, and you can have a fixed effect for different environments, but it doesn't take into account that you have different measures of standard errors or any covariances between those estimates. Well, the StageWise GWAS will do that, for example. And then I've got some features for how to think about selection response with correlated traits. So the vignette data set that I'll focus on is the close to 1,000 potato breeding clones tested over 6 years at one location that were genotyped with our potato SNP array. So here's what a snapshot of the phenotypic data file looks like. You've got a column for environment, one for your genotype identifier. We've got 3 traits in this file: yield, the vine maturity, and fry color. How light are the potato chips when you make potato chips out of them? And then you can have columns for different experimental design factors, like here's a block column. You can have covariates that you measure along the way, like the stand count, which can be used. So this gives you an idea of the kinds of complexity you can have. And there is a function called stage 1 in the software, but you can also do the stage 1 with any software you want. That's really, it's quite open. All you have to do is compute the blues and their covariance matrix, and you can pass that information to the stage 2 function, which is by far the more complicated function to do on your own. Many people would have the capability to do your own stage 1 analysis. So I didn't spend a lot of time trying to reproduce all the nice other software that can do these kinds of mixed models analysis. So my stage 1 function will allow an arbitrary number of IID random effects or fixed effects, but it won't do, for example, you can't get it to do a correlated error structure like you could in ASReml. I did create, it has internally, it calls this package called SPATS, which was developed by our biometrics colleagues from Wageningen, which I like. It does these nice 2D splines to look for spatial variation. But that's about as far as I tend to go with this stage 1 function. You can see an example of calling it here with the ASReml option, and the output is a data frame which has the environment, the genotype identifier, and the blue. And then there's also the output which has the covariance matrices, which I'm not showing here. So you take that information and you're going to combine it with the marker data. So let's look at what that looks like. That function has this read geno, and it's got arguments like the name of the input file, the ploidy. There's some very basic commands you can do in terms of filtering based on minor allele counts and whatnot. This is the dominance equals true option. If you don't want to take the time to compute the dominance matrix because you have a large data set, or if you're working with inbred lines, which is not applicable to this group, but could be for other audiences, they don't have any reason to compute a dominance matrix. So that's why it's an option. But from the output, you can run this command called inbreeding, which gives you a sense of your genomic inbreeding. We heard a little bit from Tim about potentially the importance of inbreeding for some of the analyses he's doing. You get this 2-column output, which has 2 kinds of inbreeding coefficients. So for a long time, the way people typically talked about inbreeding in genomic selection was sort of the analog of what you do from a pedigree matrix. You look at the diagonal elements of the relationship matrix. And it's not exact, it's kind of a little bit of an apples and oranges because they have different, the pedigree matrix and the genomic matrix have different notions of inbreeding, but nonetheless, there's some similarity there. But then this directional dominance model that was developed in the last 5-6 years has another way of thinking about it based on the average dominance coefficient. Now, if you look mathematically at the formulas for these things, you can see they're kind of related. So I had a suspicion that they would be pretty similar in a real data set, and they are. So here's a plot of these 2 different types of genomic inbreeding coefficients for this potato data set, and they're quite similar. The one that's actually used in the regression analysis is the one from the dominance matrix, because that's the theory that we're using here for that analysis. So what is directional dominance? Up to this point in our community, we've really only talked about the dominance models that I helped to develop 5 years ago to show how you can take classical quantitative genetics theory and then apply it for dominance in genomic selection. That was for tetraploids, and then Lorena Batista and colleagues extended that to the higher polyploids a couple of years ago. But neither of these theories account for inbreeding depression or heterosis, which is something I didn't really appreciate at the time because the literature on this was really only just emerging in the diploids at the time. And so now I've extended the models for directional dominance from the diploid up to the polyploid level, and you can read more about that in the paper that I cited at the beginning of the talk. So the software should work for all even level ploidies, although I haven't done any testing for hexaploids. The formulas should work. So if someone tries it and finds a problem, let me know. Basically, intuitively, what's going on is that it's using a regression of the phenotype on those genomic inbreeding coefficients. That's what the directional dominance part of the model is. If you just use the random effects portion of it, it doesn't actually account for that. So here's the syntax for the stage 2 function, and the argument is called non-add for non-additive effects. The model that I had presented a year ago would be equivalent to this G-resid option. That's what I'm calling a genetic residual, because it assumes that the non-additive effects are IID. It doesn't model any covariance between the different individuals. And then if you use the DOM option, that introduces this directional dominance. There's also a none option, which again was useful for some inbred line data sets that I was using to benchmark this with diploids. So those are the 3 options you have for the non-add option. In this particular vignette data set, you can see the AIC difference there. The directional dominance model lowers the AIC by about 17 points. So small, but enough to suggest that it actually is a better fit to the data. And then when you run the summary command, you get the table of the variances, and you get this column PVE for the proportion of variance explained. And to make these numbers commensurate with how we normally talk about heritabilities, the variance associated with the main effect for environment is not included in the PVE column, even though I do show it the variance column, just so you see it. But this, in this data set, the genomic heritability for the additive effects is about 34%. The dominance variance, not accounting for inbreeding, would be 12%. And then the line that says heterosis is the part associated with the regression on the inbreeding coefficients. And it's a relatively small number in this case, which is on the one hand surprising because we tend to think of, we know from experience that potato, for example, does show a fair amount of inbreeding depression. We can see that when we start to do it at the diploid level. However, at the tetraploid level, it's actually quite hard to actually achieve any inbreeding. You have to sell things for a while, right? You guys appreciate that. So there's actually very little variation in the inbreeding coefficient in this data set. So because there's not much variation for inbreeding, you can't explain variance in the phenotype unless you have variance in the inbreeding. So I think in data sets where you had more variation for inbreeding in our types of polyploid crops, my guess is you would start to see this heterosis component play a big role. And you can see that in simulations as well. All right, so I mentioned at the beginning this idea of combining the pedigree and the marker data in this H matrix. So this was originally developed in animal breeding as a way to incorporate ungenotyped individuals. And that's still a very good way of using it. However, even when you have marker data for all the individuals, you actually can see there's a benefit to doing this because the pedigree and the marker data are providing complementary notions of relationship. In particular, the G matrix we know tends to overestimate distant relationships. It doesn't have what you would call a sparsity to it. If you think about a pedigree relationship matrix, any two individuals for which you have no known pedigree connection will have zero relationship in that cell of the matrix, but you don't have any zero numbers in a G matrix. There's always a number there. So blending these two pieces of information together helps to compensate for this kind of quirk of the G matrix. So the formula is really this simple. It's a weighted average of the A and the G matrices. This representation assumes that you have genotypes for all of them. If you don't, you have a more complicated formula, which I'm not showing you here. But this blending parameter, there's different ways of choosing it. One way is to just look at the AIC for a particular data set and see what value of W appears to minimize the AIC. And this is it's not like a one call to the StageWise, but in the vignette it's just a couple lines of code to generate this kind of a figure. So this shows in the red the AIC is the W is varied. I think I did increments of 0.2 from say 0 up to 0.8, and you can see there's a clear minimum at a W of 0.4 where we get the lowest AIC, and then you can look up and you can see in the black how that's affecting the genomic heritability. And in this, I think, and the change in the what you would conclude about genomic heritability changes substantially, as you see in this example. If you don't blend the G matrix with the A matrix, you would conclude genomic heritability is only 0.35. But if you do this, you conclude it's more like 0.5, which is a non-trivial, I would say, difference in an estimate. And the reason is that the non-additive portion. So I mentioned earlier how the A matrix tends to have, it's more sparse. It has zeros for more distantly related individuals, and dominance relationships also tend to decrease more quickly for genetic distance. So actually, it's like you're actually pulling variance out of the non-additive component into what you consider additive as you start to blend more of the pedigree matrix in with the G matrix, which just begs the question, like, what's the right answer, right? And I think it just shows these genomic heritabilities you have to take with a bit of a grain of salt if you want to think about what they really mean, genetically speaking. Okay, so passing now to the second part of the analysis, we've run the stage 2 to get these variances, and we've done some summary calculations. Now we want to calculate our breeding values or our marker effects. I mentioned already that there's this intermediate command glove prep, besides the fact that it does the computationally expensive part of it, it also is the avenue for masking data on the stage 1 blues, which can be used for cross-validation. You can predict, for example, you can simulate a prediction of untested genotypes. You can simulate predictions into new environments or combinations of those two. And by using the trait option, you can also demonstrate how effective, say, secondary traits might be at making selections. You can mask the primary trait, only use the secondary trait to build your prediction model, and then you can do a cross-validation. And I've got some examples like that on the vignettes. In the blub command, you have the main thing compared to last time is this argument called what, which is just where you have a set of options. What are you trying to predict? Basically, is why I called it what. So you have three options in terms of values, which are predictions for individuals. There's additive values, breeding values, which I think many of you know in polyploids are not the same as additive values because a portion of the dominance can be transmitted into breeding value. So if you have directional dominance in your model, it will include the appropriate fraction of the dominance in that prediction. And then genotypic values is the complete sum of the additive and the non-additive. You can also make predictions of additive and dominance marker effects. And then you have some other arguments here. You have this index coefficient, which I'm coming to. You have the GWAS and core argument, which by default is 0, meaning it will not compute a GWAS score. It will just compute a marker effect. If you wanted to also calculate the standard errors of those marker effects, it's the standardized marker effects that can be tested like a z-score to basically compute a p-value. So it will go do that additional calculation, but it takes more time. So that's why by default it won't do that. One thing about the package that I emphasize, because I think it's really helpful, is that whenever you make a prediction of the value, like the breeding value, it also computes for you the reliability of that breeding of that prediction, where reliability is the squared accuracy, meaning the squared correlation between your prediction and the true value. Of course, you don't know the true value, so that's why it's only in theory that's the squared accuracy. Assuming the model is correct, that's what the prediction accuracy should be. But our model's never perfect, so you have to take these numbers with a grain of salt. The other way of doing it is with cross-validation. The problem with cross-validation is that the data you use for validation are not correct either. So neither one of these gives you the actually correct measure of accuracy, and I think both of them in practice can be helpful to give you a kind of a lower and upper bound for what maybe you're looking for. Using these kinds of results, you can illustrate some basic principles or phenomena that I teach students in graduate school about genomic selection. For example, that marker-assisted selection always has higher accuracy than marker-based selection. It's important to realize as a breeder. If you have phenotypic data for the selection candidates on top of the marker data, having that phenotypic data, combining it with the marker-based prediction gives you a better selection. It's intuitive, right? So you can see that easily from the reliability calculation. If you mask your phenotype data and you make the prediction, and then you unmask it and you make the prediction. Here's the predictions with the phenotypes is called marker-assisted selection, which has the points are way above this dashed line, which is the 1-to-1 line. If they were equal, they'd be all in that line. So, okay, let's look about the multiple traits. So the syntax is very similar. You call stage 1, and now only ASReml is an option. I don't believe SPATS, to my knowledge, handles multiple traits. So you have to have ASReml to do this with the StageWise package. And I put here the output. If you use the silent equals false, you see the iterations of ASReml, just to give you an idea of how long this takes. So for this data set of 1,000 clones with the 3 traits, each iteration took about a minute. So it's about 8 minutes to get this to converge to run the REML calculation on a high-end laptop. The output from the summary shows you for each of the 3 traits, the variances in the original units. Then it has that proportion of variance explained. And then the bottom, we have the correlation matrix. And previously, I had both additive and non-additive, or I had additive and total correlations, and it became more complicated to do that as I fine-tuned some of the theory. So all you have now is the additive correlation, or whatever the leading order was. If you don't have marker data, it's the total correlation. If you have marker data, what you're seeing is the additive correlation. So for this example, we have a pretty high correlation between the vine maturity, which is the common measure of maturity in potato, and total yield. So, and because of the way our rating scale for maturity is, higher numbers are later. This means the later maturing plants in our data set tend to have higher yield, which is a very well known, a very common phenomenon in plant breeding in general. You select for higher yield unintentionally, you could also get later maturity, which is typically undesirable. So there's different ways of trying to deal with this, but one is to use a multi-trait selection index to try and weight your traits and avoid these kinds of undesirable correlated trait responses. And particularly for parent selection, this is a very practical way of doing it. Because the analysis is a multi-trait BLUP, the index coefficients are the genetic merits. Whatever genetic merit you want to assign in units of standard deviation, those are the coefficients of the index. You don't have to do any additional analysis. For those of you who know what a Smith-Hazel index, that's actually a special case of BLUP. So by doing BLUP, you've already taken care of all those complex covariances that go into that calculation. This function I wrote called gain can help us understand some of these trade-offs in a data set like this. So here's an example of an index where I'm just going to give naively equal weights for yield and fry color, which is, I think, if you survey potato breeders, a common sentiment. We need both of those traits, and I'd have a hard time saying which one was more important. I'm going to give 0 weight to vine maturity, and because, let's say, there's no intrinsic value, although I know in practice I don't want it to be later, I don't have a specific economic value I'm putting on it. So if I do that and I run this gain command, I see in the response, I get about similar responses. This is in units of genetic standard deviation for yield and fry color, but I also get a response for vine maturity, which is what you'd expect because it's got this positive correlation. I don't want that. I want to limit, I want to have 0 response, or if anything, a negative response for vine maturity. And I can do that by using what's called a restricted index to ensure that I don't have any positive response for yield. So here's the call to this gain command with this additional argument called restricted, where you give it a data frame of the traits and what restrictions you want to place on them. And unlike traditional plant breeding methods, if you are steeped in this, typically always use equality constraints. You would apply an equality constraint, and it becomes a Lagrange multiplier in the solution of the equations. With my background in convex optimization, I realized you could actually go beyond equality constraints. You can use inequality constraints, which can have some advantages. Like in this case, I'd be willing to accept earlier maturity. That actually would be a good thing. I just don't want later maturity. So my restriction is that the gain for maturity has to be less than or equal to 0. I'm willing to accept a negative gain, which would mean shorter maturity in this case. Well, because of the correlation structure, it turns out the best solution is still a 0, not a negative. So there was no benefit in this example to having the inequality. But in the paper, I do give a toy example where I can show situations where you actually do derive some benefits in terms of getting a better optimal response. And you can see that we've had to, the price for restricting the gain for maturity to 0 is that we give up gain for yield. There has to be a penalty. So I've reduced my gain for yield down to 0.3, but I've been able to maintain my gain for fry color at 0.512. And then you can take the output from this gain command and you can plug that into the BLUP command, which has this argument for index coefficients. So you can then calculate your BLUPs for your breeding values or whatever other prediction you want to make based on this index. There's some also interesting tools for visualization in the package, which I don't have time to go into, but you can visualize some of these responses and the genetic merit vectors and all this stuff. But since Dave stood up, I'll skip to the last slide here to summarize some key things here. Hopefully this is going to help our community utilize multi-trait, multi-environment data sets as fully as we can. Some limitations, let me just point out here, it's not suitable for hybrid crops, which I know there's some sweet potato breeders that are doing some hybrid crops, but for most of us, that's not really an issue. You can't combine the heterogeneous G by E and the multiple trait features at the same time. And again, as I mentioned, it does require an AS-REMO license because I haven't found something similar that has the ability to solve REMO under these kinds of constraints for R at least. So thank you, and I'll take questions during office hours later today.